Hey, Assalamu alaikum. So today my topic is apoptosis. Apoptosis is basically programmed cell death in which a cell decides to die on its own, or we can say it is cell suicide. So basically we will study the 13 reasons why the cell decides to die. So the first example is embryogenesis. In the womb of the mother, the baby's hands are like cord shape, or the toes are like cord shape. So the cells in between these cords they die, so that they can give the shape of the fingers, or that they can give the shape of the toes. The second example are the skin cells. The old skin cells die and the new ones are made. The third examples are the T cells in the thymus. Now coming towards the morphology. For example, this is the normal cell and this is the nucleus and the DNA. So the dying cell basically shrinks, and the dying cell dead materials are packed in special bags. So they have dead materials in it. Also, the dying cells express some special kind of receptors so that the defense mechanism or the macrophages can come and eat them. So the first, the cytoplasm condenses, they shrink themselves, and they expresses some special kind of receptors that are known as opsonins. So the opsonins are identified by the macrophages and they come and eat this dead cell. Now, the pathways of apoptosis, that what paths the cell is going to have to die. So there is extrinsic pathway, intrinsic pathway, and cytotoxic CD8 TCL mediated pathway. First of all, we'll be discussing the extrinsic pathway. In the extrinsic pathway, always remember that every cell in your body has a death receptor in it, and that death receptor is activated by some death inducer. The name of the death receptors are CD95, FAS, or tumor necrosis factor receptor. And the thing that is going to induce the death is known as the FAS ligand. So FAS ligand is activating the FAS, and if the FAS or the death receptor is activated, it's going to finally activate the caspase. Caspases are basically cutters which are present in your cell, or they are the sharks with big teeth. So the caspase mouth of the shark are basically closed. So this is basically proenzyme. If the death receptor is activated, so this activates the caspase and the shark mouth is open. So now the teeth are open and now they can eat all the things that is present in the cell. So if we study the caspase are of two types, the initiator caspase and the executional caspases. The initiator caspase is the first caspase which is activated, that is 8 and 9. For example, in extrinsic pathway, the first caspase which is activated is caspase 8, because we hate our ex. Hate, from hate we get 8. So we hate our ex. So the first caspase that is activated in extrinsic, that is caspase 8. And in intrinsic pathway, the first caspase that is activated is caspase 9. In intrinsic and 9 both has in it. Now the initiator caspases, for example, if the caspase 8 is activated, so this caspase activates the executional caspases. So the initiator caspases activate the executional caspase, because these are the most dangerous caspase number 3, 6 and 7. These caspases are like mommy sharks. They are the baby sharks, and this caspase, executional caspase, is the mommy shark with very big teeth and very big jaws. So they basically, if they are activated, so they cut down the DNA, they cut down the cytoplasm, and they cut down the cytoskeleton. So everything is now cut by this executional caspases. So final thing that is going to occur is by the executional caspase. Now coming towards the intrinsic pathway. Always remember that in every cell, basically, there is a balance between the life gene and the death gene. The live gene, the X, the BCLX and the BCL2 are the live gene. That is, our X is our life. And the death gene, they are the bad people, the people who talk at our back, the bags, they are the bad people. So they are the death gene. If they are activated, the cell is going to die. And if they are activated, the cell is going to live. If BCL2 level or BCLX level is going to go down, again the cell is going to die. Now, if we see, I have drawn a cell with a mitochondria in it. So this is the mitochondria. Coming towards this mitochondria, in this mitochondria there is a jail, and in that jail we have jailers that are apoptosis inducing factor, and we have cytochrome C. These jail are basically closed with some special type of doors or pores in it. These pores are known as mitochondrial permeability transition pores. So from these pores, these two jailers can go. Okay. So they, at the end of these pores, we have BCL2, and they are guarding these pores. They are basically guarding these pores or the doors. Now just imagine if the BCL2 levels go down, so now they are not present at the doors, and these jailers, that is the apoptosis inducing factor and the cytochrome C, are going to leak, and now they are going to present in the cell. So now here the cytochrome C is going to be there in the cell. This cytochrome C is going to activate the caspase 9. Okay, so now the caspase 9, the initiator caspase is activated, and then it activates the executional caspases, the big shards, that is the 3, 6 and 7. And when the big shards are activated, so they are going to cut the DNAs, they are going to cut the cytoplasm and the cytoskeleton. So now the cell is going to die, so it's going to cut everything. Now coming towards a cytotoxic T cell mediated CD8 pathway. Here, if we consider a cell which is virally infected, so every virally infected cell, the viral proteins come and express themselves on the MHC1 receptors which are present in the cell. Now, these expressing viral proteins are identified by cytotoxic T cells. So the cytotoxic T cells are going to come here, and they are going to confirm that if this is the viral protein, so they are going to confirm it by CD8. So that CD8 is going to come here, and it is going to confirm that, oh yes, this is the viral protein. So now if this is confirmed by the cytotoxic T cell that this cell is being affected by the virus, so now this activates the CD3. So here CD3 is activated. This CD3 start causing pores in this cell, and these pores are called as perforins. Okay. Now from these pores, the CD3 start releasing some toxic substances, and they are known as granzymes. So they start releasing granzymes in the cells. Now these granzymes directly activates your executional caspase. So they directly go to the executional caspases 3, 6 and 7 and activates them. And again, now the brick sharks are activated, so they are going to cut the DNAs, they are going to cut the cytoplasm, and they are going to cut the cytoskeleton. So these are the 3 pathways of apoptosis. Do like this video if you could have understand this, and do comment, subscribe my channel. Till then, Allah Hafiz, take care.